Thank you very much. Well, welcome to our first convention. I want to thank all of you for being here, many of you for being with us for the last many months since ribbon cutting took place. I know a few of you have probably not missed a single event. You've probably heard me speak more times than you really wanted to. Sometimes literally blinding you with science. Today I'm going to talk to not only those of you who have been to many events, but also to those of you for whom this may be the first event. What I would like eventually to teach all of you is the science that I think every distributor should know. This is a complicated product that you're selling, that you're representing, and the science behind it is complicated, and nobody knows that better than me. But it's also something I firmly believe you can get your arms around, and that I think every one of you can do justice to presenting this product, Protandim, for what it is scientifically. So I'm going to review all 7 of the publications upon which this science rests and upon which this product rests. When I came into this room this morning, while it was being set up, I looked up and I noticed these banners on your left. And there's a banner for every publication on Protandim, from the first one in 2006 to the 7th one earlier this year. I didn't know they were going to be there, but it's useful for me to have them because there are elements in each of those 7 publications that I would like for you eventually to have at your fingertips. I get many requests from distributors saying, I encountered, maybe it's a physician, maybe it's somebody who is knowledgeable in science, and I tried to tell them about Protandim, and they took a quick look at the science and they said, well, this is all done in cells, or this is all done in mice. Well, I want you to know exactly what each paper was done in. Probably the most important paper was done in cells, in tissue culture, human cells, by the way. But one of the papers, the very first one, was a human clinical trial. It wasn't done in mice or rats or cells. It was done in living humans. Several of the other extremely important papers were done in animals, and I want you to know what the message is from each of those. I'm going to begin with kind of a review of what oxidative stress is, and we now know Protandim does more than just deal with oxidative stress to remedy that situation. It involves not just superoxide dismutase and catalase, the first 2 enzymes we focused on, but as I told you at Elite Academy last October, we now know that Protandim modulates more than 600 genes. In fact, at higher doses, not used in humans, as many as 4,000 genes can be affected out of a total of 25,000 genes that we have. This is not a minor little tweak. This is a major readjustment of everything that makes you who you are and what you are. So let's begin by looking at this question. What is oxidative stress? Where do oxidants and free radicals come from? And a question that, believe it or not, some people don't know the answer to. Why should we worry about oxidative stress? It happens to everybody as you get older. Maybe it's just a part of life that we can't do anything about. Well, this is the number of papers published in the peer-reviewed scientific literature since about 1960, when there were essentially 0. And in 1970, there were about 3 or 4 because superoxide dismutase had been discovered. And we began to talk about what is oxidative stress. It's now been 41 years since that discovery. And the total number of papers now available in the medical literature is more than 86,000. And you can see the growth rate of these papers. And that should be kind of reminiscent of what Carrie just showed you, because this company is taking a similar exponential growth pattern. And this is the science that underlies everything that we stand for and everything that this product was designed for. The bottom line here is that scientists do worry about oxidative stress. This is a big slice, a significant slice of the total scientific endeavors going on in this country and around the world. So this is not minimal. This is not something tucked away in one corner. This is mainstream. What is oxidative stress from a cellular point of view? Every cell in your body contains these little structures known as mitochondria. They're the engines that burn our food and create the energy that drives everything we are and everything that we do. It drives the thoughts that you think in your brain. It drives the muscles that enable you to walk and run. And in principle, the mitochondrion is not very different from the internal combustion engine that drives your automobile. It takes in food, in its case, gasoline. It takes in oxygen. It burns that fuel, and it produces energy in a useful form. One of the things that mitochondria have in common with internal combustion engines is that they are not 100% efficient. They're byproducts. Ideally, we'd like for all of that fuel to appear as water vapor and carbon dioxide. And in a perfectly designed engine, that would be the case. We haven't built a perfectly designed engine yet. Your car produces all kinds of byproducts, and your cells produce some of the very same byproducts. Specifically, one of them is this free radical, the superoxide radical. It's called superoxide because it's like oxygen, only it's super. Oxygen is what supports the combustion process. This is even more reactive than oxygen. It's really superoxygen. It's a very potent oxidizing species. It's toxic to cells. It's toxic to the parts of your car that produce it. Your cars have catalytic converters to deal with this toxic byproduct, and your cells also have catalytic converters. Those catalytic converters are called enzymes. They're called, in some cases, antioxidant enzymes, such as superoxide dismutase and catalase. They serve exactly the same function as catalytic converters on your automobiles. What happens if you don't scavenge, take care of, convert these reactive products? They can do a number of very damaging things to biological systems. They can oxidize proteins, and the example I've shown here is an antioxidant enzyme itself called PON1, protects against atherosclerosis, very important enzyme. It's inactivated by oxidative stress. And so if you have high levels of oxidative stress, the levels of PON1 circulating in your plasma go down. They go down in many, many diseases, in rheumatoid arthritis, in multiple sclerosis, in diabetics, in patients with autoimmune diseases. It's a marker of what's going wrong in this system within your body. Too many oxidants are being produced. They're damaging proteins and enzymes. They can also damage fat, lipid molecules. And when that happens, it produces something you've probably heard about, T-bars. T-bars is an acronym, but it measures oxidized polyunsaturated fatty acids. Polyunsaturated fatty acids are not bad. They're essential for life. Every food you eat has some polyunsaturated fatty acids. You use it to synthesize your own cell membranes. But this free radical can damage that biological family of molecules as well. So it can damage proteins, can damage lipids, can also damage DNA. Another category of biological molecules, and everybody knows DNA is important. That's what constitutes your genes. It's what makes each of us different. It's what gives your children characteristics that they inherited from you. Sometimes good, sometimes bad, but that's the way that goes. The same free radical can damage DNA. Again, not a good thing. So here's a third category of biological molecules that can be damaged by DNA. Damaged DNA is called mutated DNA. And the right mutations in the right genes can lead to cancer, one of the biggest health problems we face. Again, the result of free radical attack on our DNA. And the free radicals can also signal events in our bodies. Some of these are not always damaging, and in the right place at the right time, they're good. Fibrosis is formation of scar tissue. So if you cut yourself, you want that wound to heal as quickly as possible. And fibrosis helps that to come about. However, if your heart becomes fibrotic, it's medically known as heart failure. The pump quits working at its normal output. Not good. Apoptosis is actually programmed cell death. It's a way by which cells can die. At times, that's good. When you're an embryo, this process of apoptosis actually is what carves out your fingers from what looks originally like mittens. And as the fetus develops, everything goes normally, and you develop hands and fingers as you should. So apoptosis is not a bad thing in the right place. But if the cells in your heart undergo apoptosis, it's not a good thing. The cells in your brain undergo apoptosis, and you may be diagnosed with Alzheimer's disease. So all of these things require balance. Some of them are universally bad, some are bad only when the amount is incorrect. This slide was the result of work that took place throughout that previous graph that I showed you in the last 40 years. And so this was pretty much in place well before Protandim came along. We knew all of this happened. We knew what free radicals do that damages our bodies. And it was only when I looked at this within the last week and decided to put this very early slide in here to give you perspective, to give you a summary, that I realized that those 7 papers I'm going to be talking about, represented by those banners on the wall, have addressed every part of this damage that's done by Protandim. Specifically, paper number 6 over there, published in 2010, showed that Protandim prevents that loss of this antioxidant enzyme, PON1. Papers number 1, number 6, and number 7 showed that Protandim prevents this damage to lipids, to polyunsaturated fatty acids in our body that give rise to T-bars. Papers number 3 and 5 showed that Protandim prevents damage to DNA. In fact, those papers showed that Protandim supplementation could prevent skin cancers in the mice that were used in that study. Papers 4 and 6 showed that Protandim can prevent fibrosis, scar tissue that can cause a heart to go into failure. Paper number 4 showed that Protandim can prevent apoptosis, or programmed cell death. And you'll notice that in many of these things, multiple papers have addressed the same issue. And there's method to that. I think some people look at the specific disease models that have been studied in these papers, and it may look like a random selection. Why are we looking at drug-induced pulmonary hypertension? Why are we looking at intimal hyperplasia in veins that are used to bypass clogged arteries? It's because it's these same 5 processes that are involved in free radical damage, oxidative stress-induced damage throughout the body. What protects us from oxidative stress is not one antioxidant enzyme or even 2, even though that's what we focused on in paper number 1. It's an entire system of antioxidant enzymes. And here we see a tiny corner of that system. The enzymes are shown in blue on this sequence of reactions. The bad products are shown in red, and the detoxified products, or the antioxidant products that help get rid of the bad stuff, is shown in green. It's the blue enzymes that make these reactions occur. The first paper really focused only on that little part. It looked at the reactions catalyzed by SOD and catalase. A second paper, which is one of the very important ones, showed that the enzymes involved in making glutathione, you hear a lot about glutathione in this industry, are also upregulated by Protandim, allowing our bodies, our cells, our individual cells to make more glutathione. Not because the precursors or the raw materials to make glutathione are limiting. Sometimes they are. More often, it's the enzymes. It's the things encoded by your genes, upregulated by Protandim, that allow you to make more of these things. Protandim contains 5 phytochemicals, and again, the structures are not important to memorize, so you'll be relieved at that. But they come from 5 different plants. All of these plants have a long, long history in traditional medicine. People didn't know how they worked, but for centuries they knew they do work. What they didn't know was that they are regulators of a protein called NRF2 and activators of NRF2. And that's the mechanism whereby more than 500 of your most protective genes are regulated by these simple, believe it or not, chemicals produced by humble little plants that have been recognized in India and China for centuries and centuries. They activate what's called the master regulator of stress-related responses, of antioxidant enzymes. How does Protandim activate NRF2? And you hear a lot about NRF2. You're going to hear even more about NRF2 as time goes on. How does Protandim activate NRF2 in a cell? This is a cartoon that illustrates that. This purple shape in the upper right corner of this slide represents one of those 5 chemicals from plants, from medicinal plants. And so as Protandim approaches a cell, the cell actually has receptors on its surface. And my analogy here is this is like if the cell is your house, Protandim just stepped onto your front porch. And it rings your doorbell when that happens. And that causes a change inside your house. You hear the chimes or the bell. And you know something has just happened that may change events within your house. In this case, an enzyme on the inside of that cellular house called a kinase has been activated, signified by turning yellow. Well, the business of that kinase is to put phosphate groups, to chemically modify a handful of molecules that it knows how to modify. One of those is that little red oval labeled NRF2. NRF2 is being held in the cytosol, outside the nucleus where the DNA resides, by this other larger protein called KEAP1. So the activated kinase now chemically modifies NRF2, and you can see that little yellow P that's been added. That's a phosphate group. So it's chemically changed NRF2. And the protein that's holding on to NRF2 now sees this thing has changed its shape. It doesn't fit my grasp quite as well. It releases that Nerf2. So it's now free to diffuse around the cell, and it's small enough it can actually enter the nucleus, which is the vault. It's where all the plans, the blueprints for everything you are reside in the form of DNA. So this activated Nerf2 now has access to all of those blueprints, and it binds to specific genes within there. Out of 25,000, at least 500 or 600 bind activated Nerf2, and the result is those genes are turned on. Each of those genes has a switch, like a dimmer switch on the lights in your house. Nerf2 simply turns that knob. Those genes express what they encode. The products designated by the blueprint of each and every of those genes is now produced. And among those products are antioxidant enzymes, anti-inflammatory enzymes, antifibrotic enzymes, all kinds of stress-related genes that help cells, especially when they're in chemical stress, when they're in oxidative stress. It helps bring things back into balance and to provide a normalizing influence on those cells. We can visualize this microscopically. The upper three panels show control cells, cells without Protandim. And here, again, I won't go into a lot of detail, but we can stain Nerf2, and it stains red in this slide. It's a fluorescent red stain for Nerf2. We can also stain the nuclei of those cells. It's staining the DNA. And if you merge those two, you get what you see in the upper right. And it may be a little hard to see in this lighting, but what you see if you look carefully at that is the nuclei are green, and the Nerf2 is red surrounding the nuclei. It's not where the DNA is. If we treat those same cells with Protandim and now stain the Nerf2 and stain the DNA in the nucleus and now superimpose the red stain and the green stain, what you see is the lower right figure. Everything looks yellow, and that's because if you add red light and green light together, what your eye sees is yellow. This proves to scientists looking through this microscope that Protandim treatment caused the nerve to go from its location outside the nucleus in the upper row to exactly where the DNA is. That is, the Nerf2 has moved into the nucleus, and that's where it can regulate those genes, turn up all of those processes. The first paper published in 2006, I've condensed into one or two sentences for you here. This is really what you don't need to know about everything in that paper, but you do need to know this. After 30 days of supplementation, T-bars, which is oxidized lipids, declined by an average of 40%, and the age-dependent increase was eliminated. By 120 days, SOD and catalase had increased by 30%, 54% in the red cells, respectively. That's very significant. You have all seen this figure. This shows what T-bars looks like in a population of varying age. There's a lot of scatter. We're not all the same at age 40 or 50 or 60. Some of us are in great shape. Some of us are already suffering from diseases associated with aging. If you draw a linear regression line, the line through those scattered dots, you can see that there's a relentless progression as you go from 20 to 80. Your T-bars averages go up and up and up. If you look at people in this group who were taking conventional so-called antioxidant supplements that many of us took for decades, those are the points that turn from red to blue. So some of these people were taking about half a gram of vitamin C every day, about 400 units of vitamin E. And you might think they would have been better off than their colleagues who weren't taking those. But they weren't. Those blue dots, in fact, if you draw a linear regression line, fall on a line with an even steeper slope. And you've heard a lot in Time and Newsweek and on the evening news, studies that show traditional antioxidants, antioxidant vitamins don't always help. Sometimes they can even make things worse. Well, indeed, that's true. Our study confirmed what many scientists had known and had been publishing for years, that sometimes traditional antioxidants can help, often they do nothing, and occasionally they can make things worse. What happens when these same people, whether they were taking traditional antioxidants or not, took Protandim for 30 days? This is what you saw. And if you get rid of the before data and just look at the after, this is what's meant by the age-dependent increase has gone completely. That line is now flat from 20 to 80. And the 80-year-olds have no more evidence of oxidative stress than the 20, the 21, the 23-year-olds in this study. Now, we have to be careful in the terms that we use here. If you looked at the 80-year-old after a month, you could still tell that was the 80-year-old, okay? It did not reverse the aging process. It didn't reverse everything about it, but it did stop one of the biochemical markers associated with aging. Is that significant? I think it is, and I think all of you probably think it is. It doesn't turn back the clock. Throughout the month, the 3 months that these people were taking Protandim, their clocks went around every day, 24 hours worth, just like the rest of us. Chronologically, they were still 80 years old. Biochemically, they were in better shape. This shows, again, that Protandim eliminated that age-dependent increase. Another important thing, the two antioxidant enzymes we were focused on initially showed distinct elevations, increases, significant increases. And that accounts for what happened with T-bars. It was because of the contribution of those enzymes that this oxidation was held in check. A lot of people asked then, and they still ask, what's T-bars got to do with living a long, healthy life? They may say, I asked my doctor to check my T-bars, and he'd never even heard of T-bars. And that's not unusual. T-bars is used in research labs, and doctors, except for a few, don't spend time in research labs. They spend time taking care of their patients, trying to remember what they learned in medical school 30 years ago. Well, I don't mean that as a slam. That's their job, and thank goodness that they're doing it. But they don't know what goes on in research labs, and they don't know if T-bars is relevant or not. This is a paper that, there are many papers, there are more than 8,000 papers that have measured T-bars, and they measure it for a reason. It tells those investigators something important. The title of this, Serum Levels of T-bars, thiobarbituric acid reactive substances, predict cardiovascular events in patients with stable coronary artery disease. This is a large segment of the elderly population that would qualify for this, stable coronary artery disease. And their levels of T-bars, exactly what we measured and what you saw decline, predicts cardiovascular events. Cardiovascular events are blood clots in arteries. They're angina attacks due to occluded arteries that are letting only 10% of the normal amount of blood through. They're not good things. They're things that can bring you to your knees. They're things that can cause you to drop dead. They're things that can cause recurrent chronic chest pain. And all of us have family members who have suffered with coronary artery disease and with cardiovascular events. T-bars was a better predictor of cardiovascular events than cholesterol levels. Right? If you go to your doctor, I'll guarantee 100% he knows what cholesterol is. But many of those physicians don't know that T-bars